Welcome to program Bachelor of Science second year, subject Botany, semester 4, course code BOC 104, course title Plant Physiology. Unit 7, Nitrogen Metabolism. The name of this module is Biological Nitrogen Fixation, Asymbiotic. I am Ms. Marina Janice Rodriguez, Assistant Professor from DCT's Dempe College of Arts and Science, Miramar, Goa. In this module, you will be introduced to the asymbiotic nitrogen fixation. We will also see asymbiotic nitrogen fixation by bacteria and free-living fungi and asymbiotic nitrogen fixation by cyanobacteria and blue-green algae. By the end of this module, you will be able to define asymbiotic nitrogen fixation, describe asymbiotic nitrogen fixation in bacteria, cyanobacteria, and blue-green algae. You will be able to cite examples of bacteria, fungi, and blue-green algae that fix nitrogen asymbiotically. We all know nitrogen forms the basis of all life forms because it is a building block in proteins and nucleic acid. It is also present in chlorophyll, alkaloids, and cytochromes. If we see, the air contains approximately 78% of nitrogen, but the plants cannot use it in this form. Nitrogen from the soil is continually depleted by soil erosion, chemical volatilization, and denitrification. That is why nitrogen has to be fixed in such a form that it is possible for life forms, especially the plants, to take it in. Nitrogen fixation can be categorized into two ways: a non-biological method, which is also called as physicochemical method, wherein the nitrogen is fixed under the influence of lightning and thunder. Nitrogen and oxygen of the air react to form an O. The second method is biological method. Biological method can be further categorized into two ways, that is symbiotic nitrogen fixation and asymbiotic nitrogen fixation. If we see the values, we see that biological method of nitrogen fixation is more significant than the non-biological method, as the biological method fixes more amount of nitrogen as compared to the non-biological method. In this module, we will be focusing on the asymbiotic nitrogen fixation method. Talking about biological nitrogen fixation, the conversion of nitrogen into compounds which can be absorbed by the plants by biological means is termed as biological nitrogen fixation. Nitrogen present in the atmosphere is in di-nitrogen form. Therefore, the microorganisms which fix nitrogen are called diazotropes. Coming to asymbiotic nitrogen fixation. The fixation of free nitrogen of the soil by all microorganisms living freely or outside the plant cell is called asymbiotic biological nitrogen fixation. Now, these organisms may be aerobic or anaerobic. They can be bacteria, fungi, or blue-green algae. Studies with radioactive isotopes of nitrogen have indicated that several other organisms also fix atmospheric nitrogen. Nitrogen fixation by bacteria. The soil contains special type of bacteria called as nitrogen-fixing bacteria. They convert the free nitrogen of the soil into soluble compounds which are absorbed by the soil plants. These can be categorized into three groups, that is free-living aerobic nitrogen-fixing bacteria. Second category is free-living anaerobic nitrogen-fixing bacteria. And the third category is free-living chemosynthetic bacteria. The first category, free-living aerobic nitrogen-fixing bacteria. If we look at the title, it is self-explanatory as they are free-living, they are not associated with any plant. They are aerobic in nature, that means they require oxygen Free-living aerobic nitrogen-fixing bacteria can be further divided into photosynthetic and non-photosynthetic. Examples of photosynthetic bacteria include Chromatium and Chlorobium. Non-photosynthetic bacteria include Azotobacter, Azomonas, Dirksia, Bijerinkia. Going to the next category, free-living anaerobic nitrogen-fixing bacteria. These bacteria, even though free-living, they are anaerobic in nature, meaning they will not require oxygen. These two can be categorized as photosynthetic and non-photosynthetic. Photosynthetic free-living anaerobic nitrogen-fixing bacteria include Rhodospirillum, and non-photosynthetic bacteria include Clostridium. The third category, free-living chemosynthetic bacteria. With the name, we understand that these bacteria can break down simple inorganic molecules for their energy requirements. These include desulfovibrio desulfuricans bacteria. Nitrogen fixing by free-living fungi. The yeast that is present in the soil is also called as soil yeast. A few species of the soil yeast is involved in nitrogen fixation. An example of it is Pulilaria fungus. It is involved in nitrogen fixation. Nitrogen fixation by cyanobacteria and blue-green algae. Among the cyanobacteria, unicellular, filamentous non-heterocystis and filamentous heterocystis fix nitrogen independently. Cyanobacteria grow mainly in the crop fields. The site of nitrogen fixation in the cyanobacteria is the heterocyst because the enzyme nitrogenase required for nitrogen fixation acts under anaerobic condition. This is a picture of nostoc. And here is a picture of Anabina, and you can see the heterocyst which is marked there, which is very different from the rest of the cells. Unicellular and non-heterocystis cyanobacteria fix nitrogen by specializing some cells which have reduced levels of oxygen. Typically, they fix nitrogen in dark and photosynthesize in the light. There are about 40 species of blue-green algae that are found to be fixing nitrogen into nitrogenous and ammonium compounds. Most of these bear thick-walled heterocysts, which are the sites of nitrogen fixation. Examples are Oscillatoria, Nostoc, Schinotema. These are my references. Thank you.